I had no interest whatsoever in autophagy when I started in the topic. My postdoc, I was working on protein targeting, and in particular, how did proteins get to the vacuole in yeast? And when I started my independent position, I was going to pursue the same topic. And I happened to start working on a protein called aminopeptidase 1 that appeared to go to the vacuole by a different mechanism. Most vacuolar proteins go through part of the secretory pathway. So I thought this will be my particular area, this alternative vacuolar targeting mechanism. I started to work on this pathway, and we got mutants that were defective in delivery of aminopeptidase 1 to the vacuole. I asked for people who were working on anything affecting the vacuole to send me their mutants so we could see if they overlapped or not. I asked for mutants from 2 labs that had just started to identify mutants in the autophagy pathway. Complete overlap. Well, I guess we're working on autophagy. So it was sort of this stumbling into a field that turned out to be very important. We were coming into autophagy from this aspect of biosynthesis, which is a very unusual aspect of the autophagy pathway. In fact, by and large, it is a degradative pathway. Cytoplasm to vacuole targeting pathway that we studied overlaps essentially completely with autophagy and just adds on a couple of components that make it selective. From that moment on, I was viewing selective autophagy as being particularly important compared to the non-selective pathway that most people were thinking about. And that's one thing that led us to start to look for more of these receptors. Well, how many pathways use a selective type of autophagy? So we started looking at mitochondrial degradation, at peroxisome degradation, ER selective degradation, indeed found that each of these has some aspect that made it quite selective. In that sense, because we were coming from a little different direction, we suspected quite early on that it was more than just a general degradative pathway. We knew in one level that autophagy was important in terms of cell death. The inability to carry out autophagy would cause cells to die under stress conditions. This was one of the mechanisms, in fact, that allowed us to clone the genes that complemented our mutants. So we found that autophagy mutants were very starvation sensitive. Yeast, wild-type yeast, are pretty resistant to starvation. I think of that as being due to the situation in the wild, meaning if you have a yeast on a grape, for example, there's a lot of carbon there, but nitrogen could be quite limiting. So they have to have evolved to adapt to conditions of low nitrogen. We found that a wild-type strain, if shifted to starvation conditions minus nitrogen, could actually live for 30 days. I mean, it could be longer. We only went 30 days and then stopped. But they do quite well. As if you have an autophagy mutant, depending on the particular mutant, they'll die within even as quick as 2 days. So that's a huge difference. And we relied on that to clone many of the genes, most of the genes, in fact. We would take the mutant strain, transform in a library of yeast DNA plasmids, and then grow the transformants up and switch to starvation conditions. And then just let them grow and take, or not grow, let them try to grow and take samples out every few days. And when you got down to the point where there was almost nothing left that was living, the ones that were alive had the gene that complemented the mutation. So it's a very simple method for cloning the gene. You just sort of do the transformation, let them grow, starve them, and walk away, and come back some typically week or in some cases 2 weeks later, who's left. So we did know in that sense that autophagy was important for starvation conditions, for survival. And it was not until people working in mammalian cells much later showed that too much autophagy would actually also be deleterious, perhaps due to overeating of the cell. But it made sense. However, it was quite interesting, of course, to see it demonstrated experimentally. Too much is also not good, as well as too little. We started with the cytoplasmic vacuole targeting pathway, which was a selective or is a selective type of autophagy. And for that reason, we were thinking about selectivity. And the next organelle we actually worked on was peroxisomes. And I just mentioned that first to lead into mitophagy. So we wanted to see, okay, is it possible there's selective organelle degradation? Because that's a little different than the CBT pathway, which is taking individual proteins that were oligomers into the vacuole. So here we wanted to know if you could target organelles. Then started to look at mitophagy. One of my former postdocs in particular, Tomotake Kanki, was very interested in mitochondria. He had done some work in mammalian cells of mitochondria and wanted to see if he could identify this selective mechanism. Mitophagy is a little bit more difficult in yeast because the turnover is not nearly so huge. Yeast always want to have mitochondria, whereas they don't rely on peroxisomes very much. They do rely on mitochondria. And so the turnover is not nearly as dramatic. Nonetheless, we thought there must be a mechanism for this to happen, and we were able to show that it does occur. And after going through the entire knockout library, which was now available at this time, Tomotake was able to identify a mutant that showed an inability to degrade mitochondria. And that turned out to be a mutant that encoded the receptor ATG32. So that was very exciting because I think that was the first organellar receptor that was identified. And that led the way to additional studies, of course, that our lab and others undertook studies to identify receptors for different organelles. Mitochondria, of course, are very interesting organelles, partly because of their central role in supplying energy, but also because defects in mitochondria are associated with certain diseases, such as some forms of Parkinson's disease. And again, that's partly where Tomotake Kanki was very interested in human disease. But it's always nice when you're working with yeast if you have a connection with human disease. And autophagy indeed gave us that connection, in part because, as you know, defects in autophagy are associated with a wide range of diseases. And in fact, some of our more recent work has been done in conjunction with labs that are much more translationally oriented. For example, labs that are working with patients or samples from patients. They will get sequence information, find a mutation either in an autophagy-related gene or one that might interact with the pathway, such as a vacuolar type ATPase. And because of that conserved nature of the pathway from yeast to human, we are able to still do the experiments that we've always done, looking at activity of autophagy, the effects of these mutations. But now we're looking at a mutation that's from a patient that might be conserved in the yeast protein. And that brings it that much more into the realm of disease application. It's not that we have cures at this time for these types of diseases, but at least you can identify the cause of the disease and know, well, don't waste time, don't put this patient through this kind of treatment because there's no point in it. We know this is the cause, and if there's anything that can be done, of course, that will be done. If not, at least you know where to keep doing the research to hopefully lead to treatment. I applied for a faculty position. I specifically did not apply in general to medical schools because I knew that would not involve much teaching. And in fact, the course I wanted to teach was introductory biology. Introductory biology was one of the courses that I enjoyed as an undergraduate. I enjoyed it tremendously. And that's one of those courses where I looked at the instructor and said, wow, I would really like to do that someday. Indeed, when I started out at the University of California, Davis, my classes were generally 300 to 450 students. And the first several years, I taught through standard lecture format, because that's all I'd ever seen as an undergraduate. So it was working fine. But I started to realize that something wasn't right about it. I realized that there's a big difference between someone listening to me and understanding what I'm saying and that person actually learning the information. So you have to do the experience yourself. You have to gain those facts through actually your own learning process. And that led me to think of how could I get that to happen in the classroom. Every day in my class, we start off with a reading quiz, which is a short, simple quiz that's just meant to test basic knowledge. You have the basic skills to do some problems, apply them. And then when we do the answer, it's not what is the correct answer per se, it's how did you get to that answer? Because that, of course, is the more important part. What's the thinking process? They're constructing the knowledge themselves by doing the problems. Well, I'll answer in a couple of ways. Because first, in a somewhat non-serious way, because one of my favorite topics that I like to read about for casual reading is time travel. And so I've occasionally thought, wow, if I could time travel, what would I do? And one of the things would be to go back to see myself, say 20 or so years ago and say, okay, listen up, there's this pathway called autophagy. And you're going to want to clone these genes and to have that foresight and just start off the whole field a little bit earlier and just look like I was an amazing person. Wow, this guy really had the foresight to launch into this field. But that's the fanciful part. The aspect that's serious is, well, how did I get into the whole direction that I went in? I mean, aside from stumbling into it, some of my students, of course, will ask me, what field should I go into now that I'm finishing up my degree? It's going to be the next big topic. And of course, I don't know. I didn't even know that autophagy was going to be a big topic. I don't have that kind of ability to look ahead and say, here's what you should really work on. My own career mostly followed a path of what I enjoyed the most. In graduate school, I truly, by and large, loved being there. I enjoyed the experiments. I looked forward to going in every day. At the end of graduate school, I tried again to think of, okay, what's my next direction? And one of the topics I'd enjoyed reading about was protein targeting. Now, my point in part is I wasn't thinking ahead saying, oh, protein targeting will be a great direction when I want to get a faculty position. I wasn't thinking that far ahead. I was just thinking, I enjoy this topic. I want to learn more about it. I want to do some research in it. So in my postdoc, I work on protein targeting. So my point is that I still fall back on that aspect. When people ask me, what should I do? I say, do what you enjoy. Because it's very hard to go in day after day doing these experiments, most of which will not work if you don't enjoy it. If you do, then all the things that don't work, you forget when you get that result that works. So I would say, don't worry as much about being strategic. A little bit of strategy helps, but really focus on doing what you want to do, and that will carry you through a lot in your career goals.
I would like to thank you very much for this amazing opportunity.
It was a pleasure to be here with you talking, and thank you.
Thank you. Thank you very much.
With that, it was a real pleasure to meet you. Thank you, Professor Dan.
Again, you're very welcome, and truly it's been a pleasure and an honor. I'm so thrilled whenever students contact me and say they want to talk to me or hear me give a talk. It's yes, of course. I'm so thrilled that you're interested, that it's just my pleasure to spend that time with you.